So the answer to the two questions is very simple. An example of analogous structure will be the wings of the bird and the wings of the insects. And the example of homologous structure will be the forearm of the mammals. You can compare the forearm of a human being with forearm of the dog, of a whale, and of a bat. Well, since we talked about it for 3 times, that means you'll see this in the exam. What is another example from last week's lecture? For the, what's the example? What's another example from last week's lecture? Oh, for the homologous structure. Remember what's another example of last week's lecture? It is the embryo. The embryo, at the early stage of the embryo, we all look the same. That is the homologous structure. So this is the embryonic development. As you can see that the chicken embryo looks very similar to the human embryo. You can see that both chicken and human we have the tail, and both chicken and human we have the pharyngeal pouches. So of course, human being, when we grow, the tail will become the bone, the coccyx bone. Another way to identify a homologous structure is to compare the DNA sequence. That's why we use the molecular techniques. We talk about that in the biotechnology lecture to sequence the DNA. That means to read or map out the DNA sequence, and then we compare the DNA sequence of the 2 species. If the 2 species they have a very similar DNA sequence or DNA fragment, then we can assume that these 2 animals they're closely related. And how do we organize them? We organize the animal in cladistics. Cladistics is a system that we group the animal by common ancestor. So it's a system that we group the animal by the common ancestor. So a clade, what is a clade? So it has an ancestral species and all the descendants, and it also forms different branches in the tree of life or the phylogenetic tree. And to construct a clade, it helps us to classify, also to identify the pattern in the evolution. And cladistics, it is based on Charles Darwin's concept, descent with modification from a common ancestor. To identify clades, we have to be able to identify in-group and out-group. In-group is basically the group of species we want to analyze. Out-group is the species or group that we know that it is diverged or outside of the they're different from the in-group. An example is here. All these 3 they belong to mammals, and this one belongs to reptile. So we consider this is an in-group. They share for these 2 they share the long gestation, and for this one they share the mammary glands. All 3 share the mammary glands, but these 2 share the long gestation, and they are different from the outgroup iguana reptile here. that iguana reptile does not have mammary glands. So the classification of animal, we are still working on it. It is the phylogenetic tree is just a hypothesis. Linnaeus, remember we talked about the Linnaean system. He is the one that who developed the binomial naming system based on the Latin words. He divided all life form into two kingdoms, animal and plants. So you are either animal or plants. So it's very simple. But we have been using his system for 200 years. In the mid-1900s, we divide the two kingdoms into 5 kingdoms. So we have the prokaryotic kingdom, and then we divide the eukaryotic kingdom into 4 other kingdoms. But in the late 1900s, in addition to the kingdom, we have 3 domains developed on top of it. So 2 of the domains is called bacteria and archaea, and then 1 domain is called eukarya. We talked about it last time when I asked you to memorize the all this domain, kingdom, phylum, and such. So they said that eukarya, the domain eukarya, divided into many kingdoms, but we still trying to figure out the exact number of kingdoms we have in the eukarya. So we have domain bacteria, domain archaea, and then domain eukarya. And then under the domain eukarya, we have kingdom protists, which can have multiple kingdoms here. Protists, they are the unicellular organisms. And then we have kingdom plantae, kingdom fungi, and kingdom animalia. So remember last time we talked about 3 examples of the natural selection. One first example we talk about is the using pesticide in the farm, and the second example is the using antibiotic over prescription of antibiotics. And when you use pesticide in the farm, you create pesticide resistance bugs that will not be responsive, or they will not be killed by the pesticide. For over-prescribing antibiotics, you have the superbug, the bacteria that is resistant to antibiotics. Now, at the end, I also talk about the third example, the extinction of dinosaur, and then followed by the rise of mammal. It is this case here, evolution. So we have mass extinction, after mass extinction, and then the new species becomes a survivor. Actually, the survivor becomes the new species, I should put it this way. Survivor becomes the new species and occupy the new habitat to fill the community role vacated by extinction. So you see that these 3 examples, they are basically follow the same logic here. So the fossil evidence tells you that the mammal species, they increase dramatically after the extinction of the dinosaurs. So mammal and dinosaur, we may overlap for some time, but during the overlapping, dinosaur is the one that dominant species on the planet. But after all the dinosaurs died, then mammals, we populate the earth. So the question is, the mass extinction of dinosaurs followed by the rise of mammal. So what, similar to the two examples we covered from last week's lecture, what are these two examples? And do you have more cells or more microorganisms in your body? In your body, including inside your body and on the surface of your body. So think about it. The first question is pretty easy. I already talked about it. The second question, well, then you have to think about it. I'll tell you the answer.